Hi class, this is Dr. Ditburner. As you can see, I have moved us into Mastering A&P and over to Physio X 9.0. I know all of you can get here now because you've all submitted lab reports already, but I'm about to take you through Exercise 1, Activity 2. So we're going to access Physio X right here. And taking us into Exercise 1 again, Activity 2. As you may have remembered from the previous video, there's a pre-lab quiz and a post-lab quiz. In both of these quizzes, you can find all the answers in the overview and the introduction. So please take your time. You can get 100% on the pre- and post-lab quizzes by making sure you understand the overview. The overview is the same for exercises or activities 1 through 5 in exercise 1. The important part in these first two things, we're talking about diffusion and simple diffusion and facilitated diffusion. And this lab this week is about facilitated diffusion. And these are both passive processes versus active processes. One important figure that you can look at here is this one, and you'll notice this is the simple diffusion that we covered in Activity 1, and now we're talking about Activity 2, which is the carrier-mediated facilitated diffusion and the channel-mediated facilitated diffusion. Both of these are for lipid-insoluble things such as sugars, amino acids, and ions, things that can't get through this bipolar lipid membrane. And so they require these carriers. They do not require energy, and that is extremely important. There's no ATP used in this process. It is a passive process. So they go down a concentration gradient from high concentration to low concentration until they reach equilibrium. And here it says right here, the process does not require energy. It uses kinetic energy, which is constant motion of the molecules, until they reach an equal number on each side, which is what we call the equilibrium. So that's the important paragraph within the overview. Then next week we're going to cover osmosis and interactive transport in the following labs. So let's go to introduction. We're talking today about facilitated diffusion. We're talking about going down concentration gradients. We can use any of these words, with, along, or down. And this is just a brief explanation of what's going to happen. So please read through this, understand the concepts, and you're going to be building little membranes, the same sort of dialysis membranes, but you're going to be adding your carrier proteins into that membrane. When we get to the pre-lab quiz, remember, you can find all the answers to this again in the introduction and the overview. So go through these slowly. There's just 4 of them. I really like the experiment this week. It's brief, but I think it will really teach you well regarding the number of protein or the number of membrane carrier proteins that you're going to put into the membranes, and about concentration and what happens when you increase concentration and when you increase the number of carrier proteins. So again, you'll note that the glucose carriers display in the membrane builder is set at 500, so it's set over here at 500. Click Build Membrane to insert 500 glucose carrier proteins. So basically we're taking that same dialysis membrane we had last week, and we're going to add the glucose carriers onto it. So basically it looks like, I guess it's kind of like a little magic here as we do this virtual lab. That's it. Now we've got our little glucose carriers in there, and then we have to drag that over. And what's going to happen is this brown thing is a barrier, and it's going to drop down when we do the experiment. When you get to the post-lab quiz, again, just 4 simple questions, and you can all get 100% on that. And the review sheet this time, again, this says, explain one way in which facilitated diffusion is the same as simple diffusion, and one way in which it is different from simple diffusion. So you have to remember back to our previous lab that we did in week number 2 on simple diffusion. And another good way to answer that question is go back to your overview and read the difference between simple diffusion and facility diffusion right down in here. The next two review sheet questions have to do with how you answered during the experiment, your answers, and show me and explain to me. Don't just say, I answered it wrong. No, it's okay if you answered it wrong in the experiment, but explain what you learned. A few of you did a fabulous job last week explaining what you learned and why, and that's what I want to see in these answers. Very brief. I think this one should take you less than half an hour depending on the base of knowledge you have. Again, save it to a PDF onto your computer and then upload it into your into the Dropbox. Good luck.